This video is to help you with the evolution project, the mutant dinosaur. This is what your initial picture, if you were looking at the requirements, it says order of progression, draw the original dinosaur fossil. I provided you a list of dinosaur fossils that you needed to create. This is an example of the draw original dinosaur. Some of the requirements that were listed is the picture needs to be large in size, detailed, complete, and include a title, just like this. The drawing individual bones for each fossil and label the mutant phenotype. Okay, I haven't done that yet because I haven't created my second drawing. My mutant phenotype will be in here later because this is the initial drawing of the dinosaur that's going to be mutated. So this is an example of the initial dinosaur. This is page 2 of the packet. The top part you need to figure out, answer the questions, and ask you about dinosaur background, so you have to research that on the internet. If I'm looking at this page here, we're now looking at the chromosome mutation. Read through this, but what you're doing is this is your original chromosome here, and then it's telling you here that you need to make a mutated chromosome. So my suggestion is to look at the different type of mutations on the internet and see which mutation you want to use. So some of the easy ones are insertion and deletion. So in this case, our example shows that this person used, I'm going to make my little pointer a little smaller. They did A, B, C, A, so they did insertion. D, E, insertion, right? So in their mutated chromosome, they did insertion mutation. And that's all you had to do for that part. Okay, this is page 3 of the packet. We're going to focus on the original DNA of the dinosaur. If you look at this right here, we're going to talk about this real quick. Right here, they tell you which codes you cannot use. Each one of these lines, where the dash, this slash is, are codons. Those codons tell you what amino acids you have. This is found out on the table that was either like a box that had position 1, 2, and 3, which goes to 1, 2, and 3, and then you found your codon in here, your amino acid in him. Your amino acid was defined in there, and that's how you know these different letters. Now, let's go back up here. So each one of these spaces should have 3 letters in each one. 3 letters in each one. These are the codes that you cannot use because they mean start and stop. Now, they have given you the start and stops, so that's why you don't have to add them within your code. They gave you this start, this stop, and when you put the code that goes with this in there, you'll see when you go from DNA to mRNA, you'll see that that's what you get, and that is a start codon as well. So let's find out how we ended up getting the code that I have for DNA. I made this up. I just came up with what I wanted to say. I just made it up. Let me try again. I didn't do this, and I didn't do this one. Those were given on the paper. The rest of these, I just made up a 3-code sequence. The letters I could use were A, T, G, and C. because that stands for DNA. Now, for mRNA, I use the code here to determine what it says here. Because if we remember, DNA has a base pair. A goes with T, G goes with C. Now, because I'm going from DNA to mRNA, my code is now A, U, G, C. So every time I have an A, I'm going to put a U. Every time I have a G, I'm going to put a C. Anytime I have a C, I'm going to put a G. Anytime I have a U, I'm going to put an A. Okay? So, in the case of what we're doing right now, we're looking at the mRNA, making the mRNA strand based off the DNA strand, because DNA can't leave the nucleus, but mRNA can. So, if I look at this right here, TAC, which is a start codon, I can only use it at the beginning of the sequence. So, T goes with A goes with U, C goes with G, because I'm writing mRNA, not DNA. That's why this is a U. Let's do the next one. The next one is ACC, or AAC. A goes with U, A goes with U, C goes with G, and so on and so forth. So be sure you're using the proper sequence for both. Again, let's just make sure the code that you can have, the letters you can have for DNA are A, T, G, C. It doesn't matter in what order you put it here, but when you come to take it down here into the mRNA, convert it into the, make it mRNA, you have to use A, U, G, C. Okay? So if you're unsure about that, I would definitely look up a video to help you. I'm sure we have videos from when we studied it in the past, but I think that's a pretty good, helpful tip. We'll go into a video that explains how to find the amino acid here in just a second. Okay, so now we're going to look at how to determine which amino acid we have by looking at the mRNA sequence. So if we look at AUG, notice 1 and 1, you're just going to match them up, AUG. So if I look at the codon chart, I'm going to look at A, and I'm going to look at U, and I'm going to look at G. And if I'm looking at A, it means I'm looking at this box. If I'm looking at U, I'm looking at this box. And then if I'm looking at G, I'm looking right here. Let me erase that and highlight differently. I'm looking right here. A, U, G. Alright, so let's look at the next one. The next one is LEU. So we're looking right here, UUG. If I'm looking at U, I'm looking at the whole box. If I'm looking at U, I'm looking at just this box right here. And then if I'm looking at G, I'm looking right here, LEU. Let's do one more, and then we'll talk about the bottom of the page. Okay, the next one is GGG. So I'm looking at G, which is this entire box. I'm looking at G, which is just this box. And then I'm looking at G, which is GLY. Okay, so this is first position over here. First letter, second base, third base. The base is the letter, so that's how you know. And that is in the packet on the front page, this codon table. Alright, so then let's look at the mutated gene. You're going to use the same DNA strand you had above, up here at the top, that we can't see right now. You're going to write the same DNA strand, but in it, you're going to make some mutations and circle them. So I've made, I think, 5 mutations. So the strand is all the same, just write the same strand you had up there, write the same strand you had for mRNA here, as you had here, and then go in and just change a few letters to another letter. Then what's going to change is down here. So if my change was, let me erase this here. Actually, I think I didn't circle a couple of my mutations. I had them circled, it looks like. So I have these 2 that changed. That one, and there, and there, and there, and there, and there. So I have 2, 4, 5. So if I look at the number underneath here, it's going to tell me exactly where I need to change those mutations down here in my amino acids. So I can copy and paste this down here, and I'm going to change my mutations where they are. So I wrote them down here. I have my number 3 was changed, 6 was changed, 8 was changed, and 12 was changed. So my amino acid sequence up here can be written identically down here, but changing those 4 different amino acids and using the codon table to change it. Just that easy. So don't make more work for yourself. The mutation is your own choice, which one you mutate and what you mutate. That is totally your decision. And just make sure whatever amino acid sequence you choose, that you don't pick the same amino acid to go into your box, into your space down here. That when you when you make the mutation, that you're getting a different amino acid. So that's my number 3. That's my number 3, right? Those are 2 different amino acids. You need to be sure that your mutation is a different amino acid to actually make a mutation. Okay? Okay, this is the last page of example. The pages in between where you're doing the questions for your habitat and your environmental questions. Those are all on you to figure out just by reading and determining your answer based upon your own dinosaur that you chose. Now, this is the pedigree. Up here are the rules at the top. You see all the rules up here. The first thing you need to realize is that the genotype for your animal is whatever your mutation. This is my mutation. And the mutation, this is telling me that it's a dominant trait. And we see more dominant traits than we do recessive. So you're introducing this dominant trait when the original dinosaur was recessive with this. When you started out your pedigree, they give you some rules right here. You need to read those yourself. But the most important one is the top one where it says some of the offspring that have the genotype homozygous recessive, they don't reproduce. So in the end, you should see more dominant than recessive like we just said. Okay, so let's look. Yours should not look exactly like mine. It's made up. There's nothing guiding you other than the directions up here. It's not like there's a right or wrong. The only thing you need to consider is what is right is you should see more of your trait at the end. So I'm going to zoom in just a hair. Hopefully this works and you can see it. They gave you this one here. They gave you the alleles here. Let me highlight it. Maybe that'll help better to see. They gave you the alleles here. They gave you this box, this one, this one, this one, this one, this one, this one, this one to start off with. you had to recognize that you had to color in this one and color in this one. You also needed to label it, which I obviously forgot to label this one, but it would be this, right? So when you build yours, which comes along here, you have to build, they gave you the 0, which is the parent generation, then your F1, you have to do the F2, F3, and then the 1000 generation. So 1000 generations from this guy here, what's going to be the case? So what I did was I started out with what they gave me. Oops, didn't mean to do that. Started out with what they gave me, and on there I went ahead and said, okay, these two parents here had kids. 1, 2, 3 kids. Of those 3 kids, 2 of them have the trait that I mutated for, right? Then one of them got married and they had kids, and their kids had the mutated trait. Then that one got married and had a child, and it was mutated. This child married this man, and they had 2 kids, one which had the mutated trait. Now let's look at these 2 here. This was the original, these parents up here. This is their child. And then they married a normal person, but they had 2 children. One had the dominant trait. That person married someone with the dominant trait, which is my mutation. They had 3 children, and 2 out of the 3 had the dominant trait. And then, let's see here. We've got, I can't see it now, but and this shouldn't be there, because these 2 are married, other than they married their brother and sister, right? Ooh, that's scary. Possible in dinosaurs, I suppose, so we'll leave it where it is. And then they had an offspring that had the dominant trait. So on and so forth. Now, the last thing we look at is, what kind of outcome did we have? What kind of prediction should I have? So if I'm looking at the genetics of it, my original, if I had a homozygous dominant trait and a homozygous, or let me try that again. If I had a heterozygous dominant trait and a homozygous recessive, I have them having 50% of their children having the possibility of either genotype. Now, if I get into the position where both parents are heterozygous for the dominant trait, it's a 75% chance they would have the dominant trait. So in the end, I'm looking at the majority of my offspring having that dominant trait. And what did these guys all have in common? They shared the recessive trait, but the dominant trait is more dominant, so it shows up more often. So this generation still holds on to the recessive trait, just not very much. The dominant trait definitely is more present and will continue to pass on because it is a dominant trait. If we look down here, I have 9 out of my 12 offspring, or generation, my 1000th generation. Only 3 out of the 12 have the homozygous recessive trait, and 9 out of the 12 have my mutation. And this should help you get through the paper. By this time you should have it finished and turn it in so we can get this project completed.